Well, good afternoon, everyone, and welcome to the front row. My name is Jamie Williamson. I'm the Executive Vice President here at Scripps Research. And it's my pleasure to introduce today Professor Michalina Janiszewska, who is an assistant professor on our Florida campus. I'm sitting here on the La Jolla campus, so we have a true bi-coastal event this week. And Michalina is going to be telling you about her exciting, groundbreaking work in brain cancer research. So Michalina got her bachelor's and master's degree at the University of Wrocław in Poland. And then she did her PhD at Lausanne at the University of Lausanne, which is in Switzerland. She did an amazing postdoc at the Dana-Farber Cancer Institute, which is part of Harvard Medical School, where she was a postdoc and an instructor. We were fortunate to recruit her to the Florida faculty in 2018. She received this prestigious NIH K99 Pathway to Independence Award, which is a grant to help faculty establish their labs. And she was recognized as the American Association for Cancer Research Next Gen Star in 2019. So she is a rising star in the application of genomic methods to understand and characterize heterogeneous populations of cancer cells, in particular focusing on brain tumors. So cancer is a very complex disease, and I'm sure many of you are asking, well, with all this money pouring into cancer research, why don't we have the cure? And it's really staggering how complex the biology of cancer is. So Michalina is going to give us a deep dive into some of those challenges and try to explain to you in lay terms what the challenges are to making medicines and new treatments. So today's talk is cutting edge technology to ask questions about the basis for future treatment. So one thing that I need to make clear, we had this in one of the slides, that this is not a presentation about treatments per se, but the underlying basic biology of what causes cancer and how we can use new technology to invent the medicines of tomorrow. So I'm sure there's no one in the audience who is not either been a cancer patient or has a family member or loved one who has had cancer. This is a universal presence in our lives. But we are a basic biomedical research institute, and we can't comment on treatments or give any advice about what you should do. So I really hope that Michalina will serve as a nice ambassador to the way that basic research can contribute to future medicines. And I'm looking forward to her presentation. So without further ado, Michalina, take it away.
Thank you very much for this kind introduction. And this is my disclosure. So I'll start just by giving you several facts about cancer. So I guess everybody in the audience already knows that cancer is one of the leading causes of death, accounting for almost 10 million deaths yearly worldwide. But the good news is that while the incidence of different tumors is still very high, and it seems like it may be growing, many of the tumors that previously have been very deadly are now sort of falling down in this ladder of cancer mortality. So this is the case, for example, for the breast cancer, where even though this tumor is really very prevalent, it's the second most prevalent tumor of all of the solid tumors, it's actually not as deadly as it used to be 15 years ago. And this is all due to the fact that there's a tremendous effort in research of the fundamental cancer biology. So this graph is showing you the increasing funding of the government and private institutions that was pouring into the cancer research over the last decades. And you can see that this very clearly translates into the scientific output of publications. But this is not only about just scientific papers, but actually this increase in funding, it actually did translate into a steep decline in cancer deaths. Since 1991. It's declined down almost 30%, and that accounts for 2.9 million of cancer deaths that have been averted in the US alone. And there is almost 1,500 new cancer drugs that have been approved by the FDA since 2006. So then, as Jamie said, there are so many cancer drugs, but then why don't we have a cure? And this is the fundamental question that I'm going to try to address today. But to be able to talk about that, I have to start with Cancer Biology 101. So this is a classic picture that you may see in the sort of older textbooks, where in the process of tumorigenesis starts with a normal cell that stochastically, so just by accident, makes a mistake in the replication of its DNA. And this mistake, called the mutation, can either be tolerated and not change anything. This can lead also to cell death, or it can actually alter slightly the behavior of the cell. And if that happens, then this cell will keep proliferating and it will keep accumulating additional mutations. And this can actually lead to this to the transformation of this normal cell into a cancer cell. And subsequently, if the cancer cells are proliferating, they are going to generate a tumor mass, and this is what is actually detected in the patients. So what happens with this growing mass of cells, cancer cells, is that they actually affect the architecture and the organization of the tissue in which those cells reside, basically changing the function of the tissue and the organ in which this mass of cells is arising. But it's not only about the architecture. The cancer cells have also very abnormal appearance, which actually is linked to their molecular properties. So for example, the difference between the normal cell and cancer cell is that cancer cells have much larger shaped nuclei, as shown here. They have many more cellular divisions, and in general, they are very disorganized in terms of their arrangement. There is a vast variation in size between cancer cells within every tissue, and there is a loss of normal features as well. This sort of structural abnormalities, of course, are kind of underlined by the molecular properties of all of those populations of cancer cells. And those changes are actually called hallmarks of cancer. So those are functional changes that are present in many cancer cells, and these basically allow those cells to, for example, evade growth suppression or evade limits of cellular lifespan, invade the surrounding tissues, and interact with blood vessels to promote the growth of new vessels to support the growth of the tumor. But the interesting thing is that actually, this is an oversimplification, of course. There is many more of those hallmarks, but there is also the problem of the fact that those features are not all present in the same cell in a tumor. And this is actually the problem that we are going to focus on during the rest of the talk. So just to illustrate it a little bit more, we are sort of looking at the evolution of tumor again here in this picture. So we are starting with a normal cell that can divide into two daughter cells over time. And then this cell can stochastically accumulate a mutation, and this mutation can lead to the evolutionary dead end. So basically this cell is not going to divide anymore and it's going to die. Or if this mistake allows the cell to acquire a new property, this cell is then going to keep dividing. And eventually this can lead to another mutation that can produce a different feature. For example, such as it can induce the secretion of a growth factor that supports the growth not only of this branch of the evolutionary tree, but it can also affect the other cells with different properties that are sort of daughter cells of some common ancestor of this one. So in general, this creates those kind of complex structures of cells with many different mutations, kind of branching out and differentiating from each other as the time passes. And this is exactly what makes the treatment of a tumor really difficult, because these different cells have different mutational composition, and this can just be very difficult to actually find something in common for them. And moreover, they can also cooperate with each other. So for example, this branch of the evolutionary tree of tumor will actually benefit from the growth factor A that is secreted by this branch or clone. So this can also be illustrated in a slightly different way. So I guess this may be a little bit more familiar to some of you in the audience. So if you're thinking about cancer evolution, it's not really as simple as this picture over here, where you have a single sort of species which grows and proliferates, and then at some point in time there is a new species that arises. And this species then takes over and becomes more prevalent, eliminating the previous one, and so on and so on. So this is what we call the clonal sweep, which actually basically every single clone, the new clone that arises is actually causing the extinction of the previous species. But this actually is not entirely true for most of the human tumors. And in fact, we are dealing with a situation that is much more similar to this picture, where we actually have multiple different species present at the same time and competing for the same resources. And this can be also illustrated in this picture. So at every given point in time, you actually have a mixture of different types of cells in our case, but different types of species. So this is what we call clonal heterogeneity. And basically what's important about this is that between all of those different clones that are present at the same time in a given tumor, in our case, there is a dynamic interaction, which seems to be essential for the tumor growth. So again, the tumor heterogeneity is a really big issue when we think about the treatment, just because of the fact that if we have a highly diverse population of cells, when we start treating a tumor, then there is a high probability that some cells with particular features that allow them to survive the treatment will just already be there before we even start. And then the survival of the cells is going to lead to the tumor relapse. So I'm just showing you here an actual real example of a breast cancer tumor, which has been stained for two markers, HSF1 and HER2. And the HER2 is actually a marker of a therapeutic target. So just by looking at this image, you can appreciate that there is a lot of cells that actually express, so that present this marker on the surface, so the green staining kind of surrounding the blue nucleus. But you can also see that there is a lot of regions and a lot of individual cells that actually don't have this green marker on their surface, which basically means that if we were to treat this particular tumor with HER2 targeting therapy, then those other cells that don't have this marker on their surface, they would actually survive the treatment and probably lead to the tumor relapse. So what complicates things a little bit more is that it's not only about the cancer cells, but it's not only about the diversity of cancer cells, but there's also the cooperation of the tumor with the surrounding cells of the normal tissue. So for example, tumors are very good at instructing the growth of blood vessels, at communicating with the immune system, and also co-opting some of the other cells present in the given tissue. So we are really trying to understand this whole tumor as the ecosystem. So we are trying to really understand the different interactions between the distinct populations of cancer cells, but also how they actually interact with all those other components of the tumor microenvironment. So the goal of my lab is to understand and disrupt the cancer ecosystem. And the way we are doing this is somewhat similar to the way people are studying ecosystems, such as marine ecosystems of the coral reefs. So first of all, we definitely want to know what kind of species are present in our ecosystem. And then we want to understand what is the relationship between those species and the environment in which they are living. So in our case, the relationships between the cancer cell clones and their tumor microenvironment. And finally, we are also devising a lot of different studies to functionally address the ecosystem. So try to find mechanisms that are supporting this ecosystem and try to disrupt them to find better ways to treat the complexity of cancer. So I'll start with our approaches towards classifying species in heterogeneous tumors. So in the recent years, there has been a lot of advances in generating different types of profiles from tumors. For example, a very popular approach was to resect the patient's tumors, then to lyse this resected tissue, and then generate what we call the bulk profile. So basically, this profile allows for for learning something about the fundamental biology of the whole tumor, but the caveat is that it's actually highly dominated by the most prevalent cell type within this mixture. So it's a very, very general way of actually learning something about this complex ecosystem of a tumor. However, it was very instrumental in developing many of the therapies that we can actually apply right now to different types of cancer. In the last few years, however, there has been a really big boom into profiling of single cells. So what that means is that instead of just lysing the whole tissue of a tumor, we are first dissociating those into single individual cells, and then the profiling is actually done for each individual cell separately. And this allows us to generate qualitative and quantitative measures to look at the differences between different tumors or different localizations or before treatment, after treatment. And then also it allows us to discover rare cell types, because this is really not driven by which cell is the more prevalent, but we can actually really say something about very rare clones. And just as an example, I was involved in a couple of studies utilizing these technologies. And for example, because of the single cell approach, it actually allows us to look at interactions between different types of cells within the breast cancer. So in this particular study, we were able to identify mesenchymal stromal cells, so cells that before were not really thought to play a crucial role in the development of metastasis, but we actually were able to show that these are the cells that instruct other cells of the immune system to adopt the pro-metastatic phenotype, and that basically allows the tumors to generate polyclonal metastasis that can be very difficult to treat. And on the other hand, we also had a study where we combined the results of a patient, of analysis of samples from clinical trials from patients, combined that with preclinical models in, again, a subtype of breast cancer that is called the HER2 positive breast cancer. And there we were looking at very different mechanisms of resistance for the different agents that were targeting the HER2 cells. And then what we found was that very often it's actually more important what kind of cells are within the tumor microenvironment rather than the actual cells of the tumor. And this was really interesting, and I hope that it will be in press very soon. However, right now my lab is focusing predominantly on glioblastoma, which is a most malignant form of brain tumor, which is not that frequent because the incidence of this tumor is about 3 in 100,000. However, it has a very high morbidity and mortality. And the 5-year survival for patients with this tumor type is about 6.8%. So that's really difficult diagnosis to get. And basically, this is due to the fact that those tumors are extremely heterogeneous. So you can see that even over here on this MRI image, where the tumor basically has those regions that are very diverse in the amount of the contrast agent that was absorbed by the different areas within the tumor. So one of the projects that we are running right now is looking at the organization of Glioblastoma Ecosystem. And this is done in collaboration with Dr. Oskar Sentirmai from Cleveland Clinic, who is collecting for us samples from different regions of the same tumor. And we're particularly interested in this because you can already see on this MRI image that there is a very strict diversity in this amount of contrast that this tumor is absorbing. And in particular, we are really interested in comparing the sort of regions that are more central in the tumor to the regions that are more sort of buried deeper into the brain, because these are the regions that are very difficult to resect. And if we can somehow figure out how to kill off these cells, then probably we'll be more successful in preventing the relapse after surgery. So what we are doing in the lab is we are profiling those different regions from the same tumor using the single cell approaches. And that allows us to target, to get those single cell profiles from thousands of individual cells. And in turn, that really allows us to look at the composition of those different regions. And what I'm showing you here is one example where we actually see that this particular tumor has a very distinct profile in terms of the tumor microenvironment cells. So those are different types of immune cells and also some normal cells of the brain, so astrocytes and oligodendrocytes. And you can see that the cells, that the samples that are sort of more on the surface of this tumor are really much more enriched in the macrophages and monocytes compared to the samples that are buried deeper in the brain, which actually have much more normal brain cells and some cycling cells, but they have much fewer cells of the macrophages or dendritic type. So next, we are still very interested in classifying the niches in the heterogeneous tumors, because what I've shown you before is actually not really telling us much about the spatial organization of those tumors, apart from the fact that we know which region they were taken from. And this is of particular interest in glioblastoma, because those tumors are known to have different regions that are sort of creating very different environments for the cancer cells to grow. So in GBM, there are very frequently regions with very high density of really small blood vessels that supply nutrients and oxygen to the cancer cells. But then at the same time, the same tumor can have regions very close to those regions with blood vessels, which are actually necrotic, which means that there is a lot of cell death and a very particular arrangement of the cells around this necrotic region. There is also regions with differential infiltration in terms of the immune cells, and of course, the regions where the tumor is actually invading into the surrounding normal tissue. So we think about those different environments in the sort of resource type of thing, because then the different environments translate into the different resources for cancer cells. And this is also illustrated here, where you can see those gradients of, for example, oxygen and nutrients for the cancer cells that are very different in vicinity of those vessels compared to the other regions that are further away. And this is, again, somewhat similar to what we could see in the marine ecosystem, where different species actually occupy different levels of the coast, depending on how deep the water is and where the sunlight actually penetrates. So in the same way, we're thinking that cancer cells that are sitting close to the vessels or that are sitting closer to the necrotic regions, they may have very different preferences and very different properties, just like the different species occupying those different levels of the coast. So that's why we are thinking about cancers not as if they were a mosaic of really random assemblies of different cells with very different features, but we're trying to think about them more in a sense of the night sky puzzle, where we know that pieces should actually fit together, but we still have problems to understand how they actually fit and what the complete picture should look like. And this is the reason why we are in dear need of new tools to illuminate the distinct features of cancer cells within the tissue, because that will allow us to see those difficult to discern patterns if we have the right tools. So one of the tools that we are using in the lab is the methods that I developed previously, that is called the Starfish. And basically what that method does is that it allows us to use the ultra thin slices of tumor biopsies that are shown here. And we can use this quite complicated method to detect DNA mutations within individual cells without the need of destroying the tissue. So this is how this looks in practice. So those gray circles, those are the nuclei of the cancer cells, and those colorful dots are actually our ways of marking which mutations are sitting in each cell. And this is really useful because we don't need to destroy the tissue in order to know which cell has which mutation. And that allows us to sort of deconvolute the cell localization from the imaging. And then based on this, we can generate topological maps of tumors. So those maps are somewhat similar to the elevation maps, except that it's not exactly like this because it's more of a density map. So if you think about this, it really allows us to measure the distances between the different cell types. We can, based on that, infer the interactions. So if we always find the pink cells sitting together, that probably means that they need to be together, otherwise they won't grow. And this can also allow us to identify cellular preferences and hopefully the dependencies that we will be able to exploit to generate new treatments. So we are using this approach in one of the projects in the lab that we are kind of close to completion, where, for example, we are using, again, those ultra-thin slices of biopsies, except this time we're actually taking different regions from the same biopsy, and then we put them in this small array, which allows us then to profile each one of those dots using different methods. And we can profile both the genetic mutations, but we can also look at proteins that are involved in brain tumor biology, immunology, and neurobiology. So you can see sort of the scale going down. So this is just the piece of tissue, and every single like little dot in here, every pixel, it's a cell. And what this allows us to do is to really look at correlations between different features found in distinct areas of the same tumor. And we have some really interesting findings on that. So I'm hoping that those of you who are interested in it will follow us soon. So we are hoping that this whole endeavor with assaying patient tissues are going to help us understand the heterogeneity that arises from those MRI images down through, so from the organ level down through the tissue level to the single cell level. So how about the functional studies? So as you know, in cancer research, there is many different models that people can use. So there are genetically engineered mouse models, cell lines, patient-derived xenografts, and patient-derived organoids. So each one of those models is useful for particular sort of choice of experiments. So it all depends on the question, and they vary quite a lot in terms of the cost and the time that it takes to develop those models. So in order to actually address any given question, we usually use a combination of those models because it's really relevant, and I'm going to tell you why in a second. So for example, we are using the models of 2D culture, but also 3D culture, and we are trying to understand how the different populations of cells that we find in the human tumors, how they actually interact with each other. So we perform a lot of experiments where we mix the different populations of cells together, and we try to understand what kind of differences are driven from the fact that they are actually now allowed to be next to each other in comparison to being separated. And sort of 2D cultures are somewhat simplification of what actually happens in the real tumor. So we are also using a 3D system where we can sort of mimic the scaffolds, protein scaffolds that are present in the tissue and allow the cells to sort of form a little bit more natural colonies. We also do, for example, a lot of different experiments with those 2D and 3D cultures where we can follow the different properties of those cells which change when the cells are actually mixed together. So here I'm showing you the results from one experiment where we grown 3 different populations of cells alone, and we tracked the movement of the cells. So here you can see that there is not much actually going on. So there is not many dots that you can see or many lines. However, if we mix those three populations together in an equal ratios, what we see is that they actually start traveling much further distances. So now you can see those lines in different colors representing individual cells. But basically that just tells us that having those populations together must be somehow changing their properties in terms of cellular movement. And that is really important if you think about migration of cells, of brain cancer cells through the brain parenchyma. But of course, all of those experiments in vitro are somewhat a simplification because we are very poorly reproducing the complexity of the cancer ecosystem. So we are not very good at producing in vitro cultures which would actually recapitulate the blood vasculature and the necrotic areas sort of mixed together with immune cells. It's possible to do it to some extent, but in general, there are many limitations in doing that in vitro. So that's the reason why we sometimes have to resort to using the animals. And in this case, I'm showing you how different the growth of those sort of colorful, distinct lines is within a mouse brain. So before I was showing you that they were growing as a sort of mass, a sphere with a pretty nicely delineated edges, versus here, you can see that the cells are much more happy to actually migrate through the actual brain. And we can also use animals to perform much more in-depth understanding of the sort of cell of origin question. So for example, here we are collaborating with Ed Lachika from Intelligent Imaging, and we are looking at markers that are associated with cells of origin of brain tumors, just to know where exactly are they located within the brain. So with all of those experiments, our main goal is to actually be able to eventually translate our findings into an actual therapeutic benefit. So we are hoping that by understanding more about the biology of the heterogeneous tumors, we'll be able to devise combination therapies that would target subpopulations that would allow us to control the tumor much better, or some therapies that would allow us to reduce the intratumor heterogeneity. So changing not only the cancer cells, but also changing the tumor microenvironment. And at the same time, we also are thinking that it's kind of likely that we will have to resort to what is called the adaptive therapy, where instead of trying to kill off all of the cells of the tumor, you're just controlling the growth until the tumor reaches a certain threshold, and you're treating it only to actually create sort of a chronic disease out of cancer rather than something that just blows up and grows uncontrollably. And of course, our efforts are also centered on finding what is called the keystone species. So this is again a term taken from evolutionary and ecology biology, where the species that have especially great impacts on the community could be removed, and that would result in the collapse of the whole ecosystem. So with our research, we are very much hoping that we'll be able to identify either cellular interactions or particular cellular types that will actually constitute those keystones and will allow us to target them and collapse the whole ecosystem. And of course, we are also very much interested in development of new tools to measure and report heterogeneity, because it is not something that is easily reportable to the clinician. And we are hoping that by collaborating with clinical neuro-oncology fellows, we'll be able to devise ways to actually guide the treatment of heterogeneous tumors so that we could match a patient with a particular type of heterogeneity with particular combinations of drugs that would serve that tumor the best. And of course, when I'm saying we, I mean the whole team. So those are all the great scientists who I'm really lucky to work with in my lab. And we have great collaborations with Franziska Mikor and Dalit Engelhardt, who are helping us apply mathematical tools to understand the heterogeneity of the tumors. And we have several collaborators who are clinicians supplying us with different types of tissues. And especially we're very grateful to Oskar Centermai, who is providing us with fresh tissues that we can do much more with. And of course, it's a great pleasure to be here at Scripps Research because we have really stellar colleagues and great core facilities, without which we wouldn't be able to do the work that we do. And of course, I'm also grateful for the funding to the NCI and the FCBTR. And finally, also the patients and the families who are giving their samples for research are really instrumental in this work. So we are all very grateful for their contribution. And with that, I'd like to thank you all. And then before I stop sharing my screen, I want to also invite you to the next front row lecture by Donna Blackmont, which will be on June 16. So hopefully we'll see you there. And I'll be happy to take your questions now.
Thanks, Michalina. That was terrific. I hope you all appreciated this, this really basic talk about how cancer works and the challenges that we have to making treatments. So I've been monitoring. There's quite a few questions, and I've got one from Dennis Nelson, and he asked the question: can cancer arise in fully differentiated cells, or do you need to have stem cells or intermediate pluripotent stem cells?
So I think that's a great question, which is very difficult to answer, because I think there are definitely some experimental models where differentiated cells, by sort of altering their DNA, if you can kind of push them back in differentiation and push them more towards pluripotency, then these cells actually can become sort of the stem pool of the cancer. So I think both ways it's actually possible. So probably it's not only the undifferentiated cells that are the source, but it may also be that the differentiated cells can also become the source of cancer.
So Michalina, maybe it would be good to just review, why do cells mutate? What are the sources of mutations? What, is it ionizing radiation? Is it X-rays? Is it things you eat? I mean, maybe just talk about it, and what's the mutation rate in a normal cell, and how do you kind of accumulate enough mutations to be a cancer cell?
Mm-hmm. So, all right, so let's start with the mutations themselves. So those are just mistakes in DNA, and they can arise completely stochastically. So it can be just bad luck, basically. So they can just appear out of nowhere. But of course, ionizing radiation and some chemicals can also increase the mistakes that the cell makes during the DNA replication. So when the cell divides, some of those chemicals can actually affect the whole process, and that can lead to increased mutational rates. So that's why, for example, smoking cigarettes is carcinogenic, because it actually, the chemicals in the smoke are actually affecting the mutation rates in the normal cells. But I guess the other part of the question is really interesting. How many mutations do you need to become a cancer cell? So in recent years, there has been a couple of interesting papers where people have been profiling normal tissues of aging individuals. And that actually shows that there is a lot of cells containing the mutations that normally would be considered cancer drivers in the normal cell environment. And actually, those people with those mutations don't develop cancer. So there must be some kind of interesting interplay between the mutational process and probably the tumor microenvironment. So the whole sort of immune environment and probably other sort of checks and balances within the human body that contributes to development of the cancer. So that's why sort of leading a healthy lifestyle overall is really important as a prevention tool, in a sense that if you eat healthy, if you exercise, then you're definitely going to keep your whole system in a better shape. So then in case if the mutational process starts, that the body will be able to handle it better.
A related question, maybe from Marianne Hansen. So when you have mutations in a cell, why don't they die? So what keeps them alive, and why do you have to get multiple mutations to get a cancer cell?
So Some of the mutations are actually killing cells,
Mm-hmm.
definitely. So there are some pieces of DNA, so some information for the cells that is really vital for their survival. So then if a mutation arises in those sort of pieces of DNA that are really crucial for maintenance of the cell, then the cell is going to die. But then at the same time, a little bit of mutations is actually a good thing, because that allows sort of small adjustments to the environment. So that's exactly how the process of evolution is kind of ongoing. That mutational sort of toleration of mutations allows a little bit of diversification within the tissue. So then if there is an insult, not all of the cells are going to die right away, because there are going to be some cells that are going to be able to handle the stress and basically rebuild the tissue without actually generating cancer right away. So yeah, there is a little bit of balancing act in there.
Yep, that's the Goldilocks theory. It has to be just right. Right.
Yes.
So here's a bit of a technical question. What are some of the challenges in doing this single cell analysis? So it's, a cell doesn't have very much DNA. So what's the technical challenge in amplifying the DNA so that you can analyze the genetics of individual cells?
Mm-hmm. So I mean, right now, actually, most of the single cell technologies are really centered not that much on DNA, but actually more on RNA. So that's the molecule that instructs the production of protein. And that's something that is actually present in the cell in many more copies. And this is sort of more related to the functional ability of the cell to produce those proteins. And that's why that's very, very informative in terms of the function of the cells. So this is something that people are using very widely, versus in terms of profiling mutations at the single cell level, this is somewhat a little bit more challenging. So that's why we're kind of focusing on a selection of mutations of interest, and some other people are, for example, focusing on copy number alterations. So looking at pieces of DNA that are present in multiple copies in a cell, so that it will be easier actually to get a sort of proper readout from single cells. But I guess probably it's just a matter of a few years before we are able to really amplify everything and just get sort of information about the DNA, the proteins, and metabolomic information as well from single cells, because that's a really big interest in the whole community, not only for cancer researchers, but also in immunology and many other fields of biology.
Great. So here's a somewhat related question from Monica Chonyaka. In terms of single cell approaches, how can you tell which cells are the targets of the chemokines secreted from specific cancers or immune cells? And could you also expand your topological maps using that kind of information?
So this definitely can be done when you do the single cell profiling, just because it allows you to actually look for pairs of chemokines and receptors on different populations of cells. So then you can really see like which cell is actually secreting a factor and which cells may be responding to that factor. So that's definitely possible in this single cell approach, where you're destroying the tissue and just profiling all the cells. In the mapping of the cells that we are doing, actually that's not something that is done easily. So we would have to have a very targeted approach in that sense. And I don't think we will be able to easily do it sort of agnostically without knowing what we're looking for.
Okay, here's an interesting question, anonymous, but it's asking about tumor samples from glioblastoma from long-term survivors. So is there anything different about those tissue samples and using your analysis that could be illuminating?
Mm-hmm. Yeah, so that's a great question. We didn't have yet a chance to actually acquire many of those samples, because most of the patients actually are succumbing to this disease within a year and a half. But there are definitely some individual cases of people who just go on for 20 years, right? So it will be really, really interesting to do this kind of analysis, but we haven't had the chance to look at those samples yet.
Okay. A couple people have asked about applications of machine learning So have you thought about this, or and
Yes.
been able to apply artificial intelligence?
Yeah, so that's something that we definitely are looking forward to as well, because so artificial intelligence basically allows it's super useful, especially in pathology. So basically looking at the images of tissues, which to us are very difficult to actually sort of find any differences between them. But the artificial intelligence is really, really good at finding those really minute differences. If we have the right classification, then we can really, so for example, there was a really nice study a couple of years ago where people were actually able to figure out whether a tumor was IDH mutant or wild type based on just the MRI images. So it's really neat, and there's a lot of applications like this right now where people are actually trying to just from the images of different types are trying to find the hidden patterns, and that's definitely also something that we will be looking forward to do with our essays. So right now we're sort of using a little bit more simplistic models, but yes, that's definitely something that we are very excited about.
And what about genetic predisposition? So in addition to just mutations, you might be predisposed to have certain kinds of cancer. So is there any way to kind of intersect, personal genomic information with this sort of analysis to figure out if, or even possibly develop precancer screening?
So I think in terms of brain tumor, that's really, really challenging because there is not much known about the sort of predisposition based on the familial history. So there's only a couple of mutations that are known to do that. But in terms of other tumors, I guess that could be doable. So knowing something, but yeah, that's also another thing, right? So the mutations that are kind of present in all of the cells of your body are going to contribute only in parts to the development of the tumor, which may be very heterogeneous. So it's again about the accumulation of the different events. But I guess looking at, this kind of studies will require really large sample size as well, knowing that the different people have very different genetic backgrounds. This is not something that could be easily done by a small lab like us, but definitely there are large consortia that are looking at traits that are common between thousands of patients.
Here's a question from Sherry Velasco. So Sherry was paying attention and she wants to know, how do you get the DNA out of the cells?
We don't actually. So we don't need to take it out from the cells. So basically the assays that we are applying, they are just done on the tissues. So we just need to permeabilize the tissue and the DNA, because the tissue is actually fixed. The DNA is also somewhat fixed in there, so we can just apply our assays to this fixed tissue, and we don't have to extract anything.
Okay. All right. I think maybe I'll just ask one last question as a general question. And so you talked right at the end about translation and using the information you're gathering as the basis for treatments. Maybe could you just end by giving us a sense for what that would look like, how long would it take, and in your you just started your career 3 years ago. You're using groundbreaking techniques, and what would be the fantastic outcome over your career?
So yes, that's a great question, and I think what I would really love to see is heterogeneity being included in the standard pathological reports, because we all know that there is this problem, and many of the tumors are heterogeneous, but there is no standardized way of measuring that and reporting that to the clinician, and there's also no way to actually knowing what can be done about that. So I would really love to see measures of heterogeneity being used to stratify patients to clinical trials, for example, in order to actually increase the chances of a patient of benefiting from one type of therapy versus the other one. So that would be something that I would really, really love to see. So it's probably a long way to go, but I'm not alone in this. So there's many other groups who are actually interested in that topic. So I'm hoping that this is something that finally is going to become more mainstream, and people are actually going to use that for patients' benefit.
Well, we're glad you're on our team and we're rooting for you. So thanks a lot. That was brilliant. It was a great lecture to a general audience about a really complex problem. I think you did a fantastic job. So thank you. And thanks to everyone for tuning in to the front row. Again, we have Donna Blackman coming on June 16th, and I hope to see you all at the front row. Bye, everyone.
Bye.